higher the CT value, the lower the concentration of the target nucleic acid. So, basically, it means na kapag lower ang CT value, it means na it takes lower or um, shorter or lower cycles to detect the target nucleotide sequence. So that means that since it requires lower cycles to detect the target nucleic acid, it means na it has higher concentration of that target nucleic acid. The sample has higher concentration of that target nucleic acid since at lower cycles na detect na niya ang target nucleic acid. Meanwhile, kapag higher naman ang CT value, that means na it requires several PCR or cycles. So, it requires more cycles para lang ma-detect ang target nucleic acid. So, this means na kapag higher ang CT value, it has lower concentration since need pa niya og daghan pa nga cycle in order to detect that target nucleic acid. So, understand? Gets man? So, again, the CT value is inversely proportional to the amount of target nucleic acid in the sample. So lower CT value, lower number of cycles, higher concentration na ay target, daghan ang target. Higher CT value, daghan na cycle, suggest na lower ang concentration sa target sequence dira na sample. So, it requires a lot of cycles in order to get in order to detect the target nucleic acid. So, it has lower concentration. Next slide, please. Next slide, please. Okay, so now we'll move on to the software or tools that are used for qPCR analysis. So there are actually various software tools that are used to analyze the qPCR data, each with unique features. So the popular option, so ito ang two popular options na for qPCR data analysis. So first is CFX Maestro by Biorad, which is designed for data collection and analysis. So it includes functionalities for reference gene selection, complex experiment setup, and statistical analysis. And the next one, we have here the Gen X, which is a comprehensive software that offers extensive capabilities for managing qPCR data, including statistical analysis and graphical representation of results. Next slide, please. So now we have here the advantages of qPCR. So first in our list is its advantage in real-time monitoring since it monitors the target sequence real-time. And then next is higher sensitivity and specificity. And then it's reduced in contamination risk since the entire process from amplification to analysis is performed in a single reaction tube. The risk of contamination is minimized. And then of course it has rapid results since it monitors real time, and then it also has multiplexing capability since it allows for the simultaneous quantification of multiple targets within a single tube. And lastly, it has a dynamic range. Next slide, please. So now here we have the limitations of qPCR. So first on our list is the challenges on designing primers. So designing effective primers can be challenging since suboptimal designs may lead to non-specific amplification or worse, failed reactions. And then the second limitation of this analysis, it's false positive and negative results, which is which may be due to several factors such as contamination, poor sample collection, or the presence of amplification inhibitors in the samples. And these false positive and negative results can lead to misleading conclusions. And next is its sensitivity to PCR inhibitors. Since qPCR is sensitive, it may be affected by substances that inhibit the PCR process, such as complex matrices found in clinical samples or environmental samples. And this can complicate data interpretation and quantification. Next is its cost consideration, since qPCR is more expensive compared to the traditional PCR methods. So it may limit its adaptation in resource limited settings. And then it's standard curve requirements. So qPCR requires absolute quantification. So the generation of a standard curve requires prior knowledge of the target sequence concentration. So in cases where the target is not known or the variable is not known, this can complicate the analysis. Lastly is its interpretation of results. So its reliance on CT values for quantification. It relies on CT values for quantification. So it requires careful statistical analysis, as small variations can result in significant differences in the inferred concentration. So misinterpretation of the CT values, especially in complex biological contexts, can lead to erroneous conclusions. Next slide, please. So next, we have here the innovations in real-time PCR. So first is the next generation qPCR, which is a technique that utilizes advanced sequencing technologies to enhance sensitivity and accuracy in detecting low abundance nucleotide acid sequences. And then digital PCR or dPCR, which allows for absolute quantification of nucleic acids by partitioning the sample into thousands of individual reactions. So this technology provides higher precision and sensitivity, particularly for detecting rare variants or mutations in heterogeneous samples. And lastly, the CRISPR-based qPCR, which is a technique that leverages the programmable nature of the CRISPR or Cas system to target and quantify specific sequences more effectively than conventional methods. So this enhances the specificity and sensitivity of nucleic acid detection. Slide please. Next slide please.
Wait lang, dalagcha.
Okay, so that concludes my report, my discussion on PCR and real-time PCR. So again, PCR has three steps: the denaturation, annealing, and then the extension. So denaturation, which includes breaking the hydrogen bonds. Annealing includes annealing of um, the NTPs to the template, and then the extension in annealing of the primer by the rather pala annealing of the primer to the template, and then the extension, so extension of the synthesis of the DNTP, so from the primer until um, the until it synthesize the um, complementary DNA strand. And then for the real-time PCR, so again, we have detection mechanisms. So we have the dyes, the fluorescence, the cyber grain dye, and then the Tacman probe, and then its analysis, and then the CT value. So again, a CT values is um, uh, inversely proportional to the concentration of the sequence. So, lower CT values means na higher ang concentration sa target sequence in that sample. And higher CT values means na lower ang concentration sa target sequence in that sample. So I will now give the floor to my to the next reporter.
Okay, so good morning once again. So now we are going to talk about molecular fingerprints. So imagine if every molecule in the world had a unique barcode or signature, which is a pattern that could tell us everything about its origin, purpose, and potential effects. This is the world of molecular fingerprinting, a powerful technique that reveals the hidden identities of molecules, whether in a drop of water, a piece of ancient bone, or a sample of air from a bustling city. Just as a fingerprint can identify an individual, a molecular fingerprint can uniquely identify a compound, offering scientists a key to unlocking mysteries across biology, chemistry, forensics, and environmental science. With this technique, we can predict a new drug that will interact with the human body. We can track pollution back to its original source and identify food fraud or product counterfeits, and even uncover clues from ancient civilizations that tell us how humans, plants, and animals evolved. Next slide po, please. Next slide po. Okay, so now what really is a molecular fingerprinting? Molecular fingerprints is also known as DNA fingerprinting or DNA profiling, which is a forensic method that uses a person's DNA traits to identify them. It is famous in drug discovery and virtual screening due to their ease of use and speed in substructure and similarity searches. It also entails gathering, organizing, and comparing DNA samples from crime scenes. And this approach is actually devised by Alec Jeffrey, a British biologist, in 1984, wherein DNA is 99.9% identical in each cell, with a 0.1% variation dependent on the VNTR sequence. Now, VNTR sequence stands for variable number of tandem repeats, wherein, for example, in a gene A, there has a ATGC DNA sequence, but for gene B has ATTGC, resulting in a double number of T. So that T, yan yung parang variation natin na nakakode sa ating genes as individuals. Simply, the DNA fingerprinting, it is a method for analyzing differences sa ating DNA sequence between us, and it is based on our variability and polymorphism, aiding in understanding genetic relationships, solving crimes, and identifying individuals based on their unique profiles. Next slide po, please. Nawala. Hello, nawala ang ano. Okay. So, moving on doon sa ano, next is the principles, no, or technique ng molecular fingerprint. Actually, the ideal way to distinguish an individual from other people on Earth would be to describe his or her entire genomic DNA sequence. However, this idea takes a lot of effort, it is costly and time consuming, and thus clearly not practical. So, ang ginawa ng molecular fingerprinting, they will just look for genes that are highly polymorphic. Yun yung mga DNA sequences na merong multiple alleles in the human population and is therefore different in different individuals. This becomes the principle of DNA fingerprinting, or in other words, the DNA fingerprinting works on the principle of polymorphism of in DNA sequences. So it is important doon sa DNA fingerprinting na ma-determine ang identity ng short nucleotide repeats that vary in number from person to person and are inherited. In human genome, about 10% of the entire DNA is formed of repetitive sequences. And these sequences, they are formed of few hundred nucleotides in which a specific sequence of nucleotide bases is repeated again and again without interruption and result in the formation of tandem arrangement. So these highly repetitive sequences, they can be categorized as mini satellites and microsatellites. Next slide po, please. So to distinguish these two, yung mini satellite DNA, when it comes to their repeat unit size, yung mini satellite, it is composed of 10 to 60 base pairs sa kanyang parang segment good. And then sa microsatellites naman is 1 to 6 base pairs lang. For the total length, the mini satellites, it is up to 30 kilobase pairs, whereas the microsatellites, it is usually ano ba? Parang fewer than 100 base pairs. Then for the location, the mini satellites, they are often near telomeres, whereas microsatellites, it is distributed throughout the genome. For their mutation rate, it is moderate for the mini satellite DNA, whereas sa microsatellites DNA, it is very likely to have higher mutation rate. The mini satellite DNA is applied using the DNA fingerprinting in population studies, whereas the microsatellites is genetic mapping, forensics, and paternity. And the example for the mini satellite DNA is the VNTRs or yung variable number target repeats, tandem repeats, and yung sa microsatellites naman is the STRs or the short tandem repeats. So both mini satellites and microsatellites, they are actually valuable for, again, genetic and forensic studies. However, yung microsatellites, they are more widely used due to their high variability and shorter, more manageable repeat units. So just a gist of that, yung principle ng DNA fingerprinting is to recognize individuals by, again, recognizing the highly polymorphic genes, which is yun yung DNA sequences with multiple alleles sa human population. So this short nucleotide repeats na yun, nag-vary sila in quantity across individuals and are inherited. They are also critical for determining identification. So again, about 10% ng human genome is made up of repetitive sequences known as mini satellites and microsatellites, which yun yung parang basis ng ating variations as humans. And then this make up actually the human genome. So next slide, please. Now, how do molecular fingerprinting works? So here's the list of the process. First, we have the extraction. So first dito, we will have to extract the DNA from a biological sample, which could either be blood sample, saliva, hair, or other tissue-containing cells. But here sa figure natin, makita natin is blood sample siya. And then, during extraction process, yung DNA here, ma-isolate siya from the blood sample, purifying it from the rest of the cellular material. And then, this isolated DNA will then be subjected to restriction digestion, wherein it will be treated with restriction enzymes. Yung enzymes na yon mag-act sila like molecular scissors. They will cut the DNA at specific sequences. And this process will create fragments of various lengths that are unique to each individual due to variations in DNA sequence. So here makita nyo, di ba? Yung isolated na DNA natin, parang coiled pa siya, very pa sila, compact. Nung gilagyan ng gitreat na siya ng restriction enzymes, nakat sila in various lengths and nag-form into fragments. Now, these DNA fragments, they are then loaded into a gel and subjected to electrophoresis. So this electric current here is applied, causing the DNA fragments to migrate through the gel. So here, yung mga smaller fragments, smaller fragment length na they actually move faster and they travel farther, while yung larger fragments, they move more slowly. So these movements actually result in a distinct pattern of DNA bands in the gel. Excuse me. Okay, so after that, na after the electrophoresis, gi-transfer na yung DNA fragments sa membrane. Gi-place na siya doon sa membrane actually. And then, this process also refers to southern blotting. This southern blotting, stabilize niya yung DNA fragments, which makes them easier to analyze. And then, after that, after the southern blotting, we have to hybridize the membrane with radioactive probe. And by this time, magkakaroon ng, yung membrane, nag-transferan ng fragments, will be treated with labeled DNA probes that bind to specific DNA sequences of interest. So these probes, they can be radioisotopes, fluorophores, which allows for visualization of the DNA fragments. So yung labeled probes, ito yung mga fragment ng DNA that can be radioactively labeled or can be used, can use fluorescent as well. And the DNA fingerprinting here, it is generated by adding a tag probe to the nylon sheet or yung membrane nga. And yung probe will only attach doon sa pieces of DNA that are complementary. Then, after hybridization, yung excess radioactive probe, mawash yan siya. And then after that, after the labeled probes have bound to the DNA on the membrane, an X-ray or another detection method is used to visualize the bonds, and this produces an image of the DNA fingerprint, which distinct bonds representing different DNA fragment sizes. So, after ng imaging, X-ray imaging, makita natin na yung DNA pattern. Ayan yung mga bonds, yung mga block na parang ano. And that bonds or yung mga pattern na yan is actually yung DNA fingerprint. So the resulting DNA fingerprint consists of pattern of bonds unique to the individual from whom the DNA was extracted. So itong bonds na to or DNA fingerprints, ito yung basis ng ating parang uniqueness good as individual. So it provides highly specific way to identify individuals based on their genetic makeup. Next slide po, please. So moving on to types of molecular fingerprinting, this is actually parang na-integrate dito yung yung biomolecular markers or molecular markers that we have learned from bioinformatics. So if I don't know pero baka ma-recall nyo. Next slide po, please. So first is our DNA fingerprinting with RFLP. And this one is actually parang lang siya, ang process niya ng pag-detect or how it works is parang same lang sa standard process ng DNA fingerprinting. Yun from DNA extraction, may restriction digestion, subject to gel electrophoresis, then may southern blotting, may hybridization with probes, and then detection. So it is based on actual variations in DNA sequences that affect the cutting pattern of restriction enzymes, wherein yung DNA is cut by, yung restriction enzymes, and then results sa fragments, ma-separate ng gel electrophoresis, and then magkaroon i-transfer sa membrane, which is called as southern blotting, i-incubate with labeled probes, and then ma-visualize with X-ray imaging. This is used in genetic mapping, identifying genetic diversity, disease diagnosis, and forensic analysis. And its advantage is that it is highly specific and reproducible. So marami siyang napoproduce na ano na patterns or DNA patterns. And then for its disadvantage, however, ano siya, time consuming and requires relatively large DNA samples. Next slide po, please. DNA fingerprinting with random amplified polymorphic DNA or rapid. Ito siya na biomarker is PCR based, right? And ang pinagkaiba niya is parang yung kung iisipin niyo yung standard process ng DNA fingerprinting, parang gi-integrate lang doon yung process ng PCR amplification but with random primers. So mag-start siya with DNA extraction, then I will be subjected to PCR amplification with random primers, then will undergo gel electrophoresis, and then visualization. So it uses random primers to amplify anonymous regions of the genome. So the resulting PCR products will vary between individuals. So it is used for population studies, genetic diversity, and phylogenetic analysis. It is actually simple and inexpensive, and however, it has low reproducibility due to sensitivity to reaction conditions. Next slide po, please. Another is the DNA fingerprinting with AFLP or the Amplified Fragment Length Polymorphism. So ito siya is kasama ni rapid as PCR-based na biomarkers. And dito, ang pinagkaiba lang nila ni rapid is that kasi meron siyang ligation process. So, wait lang. Sa ligation, merong i-attach na short double-stranded adapters sa ends ng restriction fragments, yung parang yung mga fragments na nakat na ng restriction enzymes. And these adapters will serve as the primer binding site. So, the process starts with the DNA extraction. Then we have here, ma-subject siya sa restriction digestion. Yun yung i-cut ng restriction enzymes yung sequence. And then magkaroon ng ligation, excuse me, ligation which is mag-add ng short double-stranded sequences. And then will be subjected to PCR amplification, electrophoresis, and then visualization. So it is used in genetic mapping, biodiversity studies again, and population genetics. And for its advantage, it generates many markers and is reproducible. And also, yet it requires specialized equipment and is more complex than the rapid technique. Next slide po, please. Another is the DNA fingerprinting with STR or the short tandem repeats. And this one analyzes the variation in tandemly repeated sequences for population genetics and forensic science. So ito siya parang masyadong short din ang kanyang process, which involves DNA extraction, then it will be subjected to PCR amplification, then electrophoresis, and then lastly is detection. So yun lang for processes lang yung sa kanya. And then it is widely again used in forensics, parentage testing, population genetics, and species identification. It is highly polymorphic and informative, requires only a small DNA sample. However, it is more expensive and time-intensive than simpler methods like rapid. Next slide po, please. Excuse me. Lastly, ito DNA fingerprinting with fish or fluorescence in situ hybridization. I will just give you the gist kasi it will be discussed din ng other reporters mamaya. Further details yun. So, For fish, nag-work siya ay it works from first magkaroon ng sample preparation, and then wherein we have to prepare a slide with a sample of cells or tissue. Often yung cells they are fixed and spread doon sa slide. Then magkaroon ng denaturation wherein we have to denature the DNA in the sample para ma-separate yung double-stranded DNA into single strands. And then we will have the probe hybridization wherein fluorescent is applied and labeled on sa DNA probes doon sa sample. Then yung probes na yon, they are complementary to specific DNA sequences of interest. Then next is we will have hybridization, which we will allow the probes to hybridize or bind doon sa target sequences sa chromosomes or within the cells. Then after that is we have to wash away any unbound probes to reduce background noise. And lastly, we will have to visualize, where we have to examine the sample under a fluorescence microscope. The fluorescence signals indicate actually the location of target DNA sequences. So, yung yung fish, it is a molecular cytogenetic technique that allows the localization of a specific DNA sequence or an entire chromosome in a cell. It is applicable to non-diving cells, allowing for broader application in clinical samples and provides spatial localization, allowing researchers to visualize the physical location of target genes or sequences within the context of a cell structure. So, with that advantage, it also has its misconceptions, wherein it has limited resolution for small mutations. It is complex and time consuming in sample preparation. It is costly and marami siyang requirements when it comes to equipment. We have it has low multiplexing capacity, and additionally, the results of this can be subjective, and there is a risk of false positives or negatives if hybridization conditions are not optimized. So the fluorescent signal will fade or magkaroon ng photobleaching, which is a potential issue, and it is not suitable for genome-wide analysis. However, despite these limitations, FISH remains a valuable method in clinical genetics, oncology, and microbiology. Next slide po, please. Now we have our applications of DNA fingerprinting, and this includes the diagnosis of diseases, diagnosis of paternity and maternity, identification of exchanged child, forensic science, and protection of legal rights. So we will have to discuss that one by one. Next slide, please. So for diagnosis of diseases, the molecular fingerprinting, it helps identify genetic mutations and biomarkers associated with specific diseases, allowing for early and accurate diagnosis. So for example here, we have here the BRCA1 and BRCA2 genes for breast cancer. So using these genes, yung BRCA1 and BRCA2, um, it can help identify the individuals at higher risk of breast and ovarian cancers, enabling preventive care and personalized treatment and strategies. Additionally, in some instances, in oncotyping in cancer, in cases of breast cancer, molecular profiling helps determine the likelihood of cancer recurrence and guides treatment, helping clinicians decide whether chemotherapy is necessary. Next slide, please. Another is our forensic science, wherein DNA fingerprinting is very helpful in detecting criminal cases like murder, robbery, rape, kidnapping, and etc. And as an example, we have here the conviction of the Golden State Killer. Si ano to siya? Anong pangalan? Si Joseph James DeAngelo. He was arrested and charged with 13 counts of murder in 2018, linked to multiple unsolved cold cases that dated as far back as 1974. So imagine, 1974 to 1986, sige na siya pamatay. Murag 12 years, tapos wala gyapon siya nadakpan. And so to find him, investigators used public genealogy databases matching the efforts with data obtained from the victim's relatives, leading them closer and closer until they found D'Angelo himself by using his discarded items such as razors and napkins to obtain his DNA, which connected him to 13 murders, 51 sexual assaults, and 120 burglaries. So the case was solved in 2020 wherein the Guardian reported yung news reported him na sentenced siya to life in prison without possibility of parole. Next slide po, please. Another is the protection of legal right. So the DNA fingerprinting, it is used to legally protect new varieties of plants and animals. So, for example, itong Monsanto versus farmers, for GMO crop protection ito siya. And the Monsanto and other biotech companies, they use DNA fingerprinting to ensure their patented genetically modified crops that are not unlawfully propagated and thus protecting their patents. So, um, ano itawag sa ko ano guna siya sa na it actually the fingerprinting, they can protect the intellectual property. And I mean experience na namin sa Corteva kung saan kami nag OJT kasi 'di ba, like naka-work kami sa seed storage, and then kanang before good magpasok, they have to check us, like i-check sa, and then paglabas ng storage na yon is i-check ulit kahit isang seed lang kasi bawal yun siya dalhin. And like dili lang siya check, dili lang siya guard ang naga-check, they have to take a video din para ma-sure na walang may gidala from inside, kasi kahit isang seed lang pwede kayong kasuhan. Next slide po, please. And then next is the diagnosis of paternity and maternity. So the DNA fingerprinting is the gold standard actually for us establishing biological relationships by comparing genetic markers between a child and the alleged parents. So for example, yung paternity testing in family courts. In cases where paternity is contested, courts often order DNA tests to confirm biological relationships, ensuring accurate parental responsibilities and rights. Like this, for example, sa paternity court, yung mga father, they try to, avoid responsibility na hindi daw nila anak nga, so the court will have to order DNA test para maano. Pero this one actually, katawanan ni siya kay grabe gid yung deny niya ang iyang anak kay kamukha man niya. Murag like basta if you can watch that pang anti-stress. Okay, next slide po, please. Then we also have identification of exchange child, where for example, yung hospital, maswap nila yung child, and that is also a case. Locally, meron tayo na ito siya sa GMA, sa Kapuso mo siya si Kasoho, na switch yung babies nila. Then, kasi yung mother nito, ang isang mother dyan, siya, first time mother siya, tapos pag na sila actually, discharge na sila sa hospital. And then, mga pag-uwi nila, doon niya na-realize na parang ibagod iyang anak. Ambot sa iya kung paunsa niya na na-feel niya siguro or ba. Na murag dili niya anak niya. Gi-test sila and negative ang baby na na yung test is negative. So, that is a proof na hindi niya yon anak. And then yung may nakasabay niya na nanganak by that time is gitest din and negative din yung sa kabila. So gi-switch na pud ulit ang baby tapos ibalik na sa ilaha. And by that ang hospital is nakasuhan siya actually. So for hospital child swap cases, when two newborns are accidentally switched in hospitals, the DNA testing can be used to confirm the identities and ensure that they are returned to their biological parents. So, additionally, one example also of this case is reuniting families post disasters, wherein DNA testing has been used in situations like doon sa 2004 Indian Ocean tsunami to reunite with families with missing children using molecular fingerprints to identify and match children with their parents. Next slide po, please. Okay, so all in all, for the advantage of molecular fingerprinting, generally, DNA fingerprints, they are unique, they are unaltered, and cannot be duplicated, making them a reliable method for individual identification. However, the limitation is that the high cost of testing can lead to incorrect results due to errors in hybridization or probing, and contamination in the sample can also affect the results. So next slide po, please. So as a summary again, ayan, one technique for identifying individual variation, our variation or uniqueness natin as individuals at the DNA level is called DNA fingerprinting. So, yung principle niya again, it works on the basis of DNA sequence polymorphism, which is a that uh, DNA sequences that have multiple alleles in the human population, therefore different in different individuals. It simply needs a tiny quantity of tissue like skin cells, blood, semen, or hair follicle roots to continue the process. And then we have different techniques ay okay. So maybe you have to question why why lima lang na techniques or biomarkers ang i-include ko, wherein from the bioinformatics subject maraming biomarkers, right? Kasi itong lima, they are the most widely used na biomarkers. And if you want to, siguro, naman na ang rapid ang pinaka-recommended. And in cases involving paternity, rape, assault, and other identification issues, DNA fingerprinting is an effective method of proving identity. And then, recent advancements have made it possible to create DNA prints using minuscule amounts of tissue. So, ayun. Next slide. Okay, so I think that covers my part in this report. Let me give the floor to the next reporter po. Thank you so much. Di na to
Maing udto everyone, kaya pa ba? React heart if kaya pa. React sad kung dili na. Okay, thank you for the confirmation. Actually, we have, I think, 29 slides left. Ayan. 29 o mga 28 slides left. And then, given na 11:38 na hindi na siya makaya kasi hindi siya makaya for the ano. So, I would like to give ano na lang, so far na i-tackle, and then for my part and for the last reporter kay kanang next class meeting na lang po. So far we have discussed, 'di ba, regarding how to study. Our report generally focuses on how to study or how to characterize or study the microbial community using varying methods. We have discussed culture-based methods. We also have discussed biomarkers and cell stains, PCR and real-time PCR, and natapos na rin natin discuss ang molecular fingerprints. We have two more left na method in studying or characterizing microbial communities, which is the fish, or which are the fish, fluorescence in situ, hybridization, and sequencing and parasequencing. So we will discuss this two for the class meeting po. And Jet, closing remarks.